Hi, we're Team VLC, and we're going to talk about our exploration of our gene Bradi1g75350.3 in B. distachyon. So to start, we needed to create a gene model for our given gene. So we did an ab initio model using FGenish software and a homology-based model using phytosome. The FGenish model gave us 8 exons, and the phytosome gene model gave us 11 exons for our predicted gene. And these can be seen in the 2 figures in the bottom. And the coding sequences of these 2 models were slightly different. Where these coding sequences are similar is in red, and where they're different is in black to compare. So we determined our final model to be the phytosome working map, and we decided this because it's a homology-based model where it is based off of a database with sequences using EST tags and contig sequences to create the model. We also looked at the phylogeny of our gene to know more information about its function. Our gene is shown in green in the middle of the tree. It has a 76% similarity to its closest neighbor, and it has orthologs of O. sativa and A. trichopoda. We were surprised to find that Arabidopsis thaliana is not on our tree because further research we did later on shows that there are some similar genes to ours. We also looked at the gene domains of our plant. We had 3 superfamilies come up in our NCBI search: ZFCHY, Ring U-box protein, and zinc ribbon 6 superfamilies. And we did research on these families. Specifically, we wanted to focus on the ZFCHY family, and that's because it is a zinc finger protein that binds zinc. And one source in particular that we found shows that in Arabidopsis thaliana, a protein similar to ours, when its expression is increased, it increases the plant's tolerance to environmental stressors. Based on our literature review, we predicted our gene's function is a zinc finger protein that helps B. distachyon tolerate changes in the environment.
So next we wanted to study our genome mutation. So it is that we blasted our sequence against the BD21-3 genome of phytosome, and we found this high impact mutation located at base pair 3598. This mutation was where cysteine changed to a thymine, resulting in early stop codon. So to further study this mutation, we created primers around that target mutation. As you can see here on our GMAP, we have a left and right primer, where our left primer is 345 base pairs away from the target mutation, and our right primer is 345 base pairs away from our target mutation. So next, we decided to DNA extract from all our 8 samples. We had 2 wild type samples and 6 mutant samples. Once we extracted DNA from all our 8 samples, we ran it through a gel. The first gel had no RNase, while the second gel did have RNase present. This is to ensure that each sample had DNA, and as you can see here, the top band is our top larger band is our DNA. To further solidify each sample had DNA, we ran them through a fluorimeter, and then they showed that each sample had DNA. Second, we ran them through nanodrop, and just to ensure that there was nuclease present and a decent amount of concentration of nucleic acids. After that, we ran all our samples through PCR and then ran them through a gel. Each gel had on gel 1, each sample had 1 band except mutants 4 and 6. This 1 band shows that our target sequence was amplified, and to further do this, we went through second gel with the ladder mix, and this showed that our 1 band has a length of 700 base pairs. Next, we ran 3 samples through nanodrop, and this is to see that each sample had more than at least 10 nanograms per microliter of nucleic acid concentration. After that, we extracted our DNA samples from the gel and then sent them off for Sanger sequencing. As you can see here, all 3 of our samples had little noise when looking at the results, and all our mutants were homogeneous, so we were able to locate our mutation. Lastly, we decided to look at the phenotypes of the first internode from our plants. Compared to the wild types, the mutants had less lignin, as you can see from the T-blue staining.
After this experiment, we researched the expression of our gene, and unfortunately, we didn't have any data on the stem, leaf, and root locations of expression. We only had data from thermocycle and the Hazen lab. However, we did from phytosome experiments, we were able to see a general trend in the table on the slide that expression of our gene is low in the leaves of B. distachyon, but higher in the roots and stems. From the thermocycle and Hazen lab information, we concluded that our gene is expressed more in colder temperatures and is minimally expressed when it is dark. We ran a t-test to compare these conditions and found that neither of the changes in expression were significant. We also researched to develop an RNA experiment. Specifically, we found a gene similar to ours in Arabidopsis thaliana, and through EFP, we found multiple experiments that changed NaCl, iron, and nitrogen concentrations and looked at the expression change in the roots of our gene. And we decided to test the effects of salt on our gene expression in stems. For our experiment, we hypothesized that the plants treated with salt would increase the concentration, their expression of our gene because it's an environmental stressor. Our treated plants had 10 milliliters of 0.15 molarity salt concentration added, while our control plants only had 10 milliliters of distilled water. And 3 hours after this treatment was applied, we took our samples.
So once we collected the stem and leaf, we did RNA extraction on all 6 samples. After that, we ran them through nanodrop to make sure there was nucleic acid present. As you can see from our results, there was. And after that, we ran all our, both 3 treatments and 3 controls through a gel to make sure that there was 2 bands present. And then these 2 bands present means that our RNA was intact.
So then we created primers, and we created these primers so that they had an appropriate amount of base pairs included in an intron within the primers, and this would tell us the difference between RNA and DNA during our qPCR reaction. So after doing qPCR, we were given the results, and from these results, we created a graph. The graph can be seen on the slide, and it shows that the treatment group has a higher fold change than the control group. Though this does appear to be a difference, we ran a t-test on this fold change and found that the change in expression is not significant between the treatment and control group. And we know that our qPCR was successful because the wild type and mutant fold change levels are different. And so though our hypothesis does appear that it could be a difference, it wasn't significant. And that's it. Thanks so much for a great semester. Have a wonderful summer. We appreciate everything you've done for us.